14.1, variation and evolution. We're going to be discussing what makes you different from your siblings. You've both got the same mum and dad, so why are you different? And we'll also look at why are identical twins not the same as they grow up. Remember from Key Stage 3 that variation can be caused by genetic differences, it can be caused by the environment, and it can be caused by both. For instance, you may have a tall mum and a tall dad, and so you are genetically predisposed to be tall yourself. However, if you're undernourished, you won't actually reach your potential. So the environment has affected the way your genes have made you grow. With the same mum and the same dad, you might expect that you would be identical to your sister if you're a female, or identical to your brother if you're a male, because you've both got information from mum and information from dad. But just like there's lots of ways of slicing a cake, you can slice it right down the middle, you can slice it right across the centre, then there's different ways of sharing your genes with your offspring. It depends on the gamete you produce. Let's consider one pair of chromosomes in mum, one pair of chromosomes from dad, and actually let's just consider one gene on that chromosome. Let's imagine that this is mum and this is dad. Here is the gene, doesn't matter what it is, it's got two types of allele. It's got a red allele and it's got a yellow allele. So when they make their gametes, there's lots of ways, there's a variety of ways that these alleles can combine. So for instance, mum might give this red one and dad will give the red one also. And that's going to create one type of individual. Mum might give this red one and dad might give the yellow one, and so on. And so as you can see, straight away here, there are 4 types of gamete that can be produced with just one pair of chromosomes with one gene that we're considering. We'll imagine 23 pairs of chromosomes with thousands of genes. In fact, when you think about it, it's amazing that siblings even look similar at all. Some characteristics are coded for by just one gene. When they're coded for in that way, you either have it or you don't. So an example is tongue rolling, doing that kind of thing. Some people just can't do it and it looks really funny watching them try, but it's because you've either got the gene or you haven't. Now, if we were representing that on a graph, we would look at a bar chart. It's as simple as that. You either can or you can't. When we look at characteristics that use a number of genes to code for them, such as height, it's not just coded for by one particular gene, there's a whole host of them that work together to produce height, then what we find is when we plot that information in the graph, there's a huge variety. It's not just like with the tongue rolling, you either can or you can't. There's a huge variety, and when we put the sample size, if the sample size is big enough, when we plot that, what we get is the normal distribution. If we're plotting 2 continuous variables like we had for height against each other, so height and weight, if I wanted to check that there's a relationship between the height of people and their weight, then the type of diagram I would use would be a scatter graph. And the closer the points are together on that line, there's more of a relationship. If they're more spread out and the line is more diffuse, there's less of a clear relationship. So on this diagram, there's very little difference. There's a clear correlation between the two. But of course, in science, we have to remember that correlation doesn't mean causation. The height hasn't caused the weight. We don't know that. That would take further investigation, but certainly that relationship shows that there is a correlation. Okay, so this is the bit I wanted to get to. It's in the book. It's one of the questions. It says, design an experiment to investigate the effect of temperature on the growth of 20 cloned seedlings. Okay, so these seedlings have already germinated. They're small, maybe two leaves on there. What are we going to do to investigate how they will grow in different temperatures? Well, the first thing we're going to do is we're going to divide the seedlings into 5 equal groups. So that would mean that there's 4 seeds in each group. And there's a reason why we're going to do that. And remember, with AQA, what they want to know is why you've chosen to do something. If you're using a particular instrument, why did you choose that instrument? What does it do for you? Well, the reason why we're going to divide it into 5 equal groups is because to plot a graph, what we want is 5 data points, because only with 5 data points are we able to really spot a pattern or a relationship. Secondly, we're going to select 5 different temperatures, and we're going to control those environments for the amount of water, the humidity, the amount of airflow, and so on. Why have I chosen 10, 20, 30, 40, and 50 as my range? Well, what I want is I want a range that's big enough that it's going to give me different results for each interval that I try, but I also want it to be achievable. There'd be no point trying to grow some seedlings at 90 degrees, I know that. There would be no point, so that's the reason why my range isn't that big, but there'd also be no point in choosing 41 degrees, 42 degrees, 43, 44 and 45, because the results I would get wouldn't be different enough. Next thing we're going to do is we're going to measure the height of the seedlings in each group. We're going to calculate a mean and we're going to record that down. And the mean just reduces the effect of any outliers. Then we are going to choose any amount of time, really, 6 weeks, 8 weeks, 12 weeks, whatever. We're going to grow the seedlings for 6 weeks, and we're going to ensure that all of the variables are kept the same, so that the only thing that's changed is the temperature. And that means that if only the thing that has changed is the temperature, then any difference in the growth of those plants will be due to the temperature. So after 6 weeks, we're going to measure the height of the seedlings again. We're going to take a mean for the group. Then what we're going to do is we're going to calculate the percentage increase in their height. So we're going to take one from the other to look at the increase, and we're going to look at the percentage increase. And that is important that we look at the percentage increase. Why? Because not all the seedlings may have started at the same height. So for example, at the moment the teachers at school are doing this competition called the Biggest Loser. Well, you've got one teacher who's 120 kilos to start with, and you've got another teacher who's 55. If they both lose 5 kilos, have they really lost the same amount of weight? In terms of their percentage decrease, they haven't. And so if you have different starting masses, different starting heights, it's always a good idea to look at the percentage change. Then what we're going to do is we're going to plot a graph, and that will give us an idea if there is a pattern there, and it will also allow us to take readings for values that we haven't tested. So for instance, would they grow best at 25? We don't know, but we can have a look at where it is on the graph. Also, when we see the maximum increase, maybe it's between 20 and 30, then what we can do, we can repeat the experiment, but we can choose different intervals. We can choose 20 degrees, 22 degrees, 24 degrees, and find out which is the best temperature for that plant. So let's have a look at some questions. What was the independent and the dependent variable in this practical? Well, the independent, before you do your experiment, is what you decide to change. So in our case, we changed the temperature. The dependent depends on the practical, therefore you do it after, and it's what you're going to measure. Now, you could say change in height or percentage increase, any of those things. What were your control variables? So back in the day, we used to say, how will you make it a fair test? All the things that you control to make it a fair test are the control variables. So the volume of water, the humidity of the air, the type of soil, all of those things. We don't have to talk about the type of seeds because they told us in the question that they're giving us 20 cloned seeds. How could you make your experiment more reliable? Same answer all the time. Repeat. You're going to repeat it and expect to get the same results. How could you ensure your experiment is reproducible? Well, if somebody else does the experiment and gets the same result. They might use a different number of seeds, they might do it in a slightly different way, but if they get the same pattern of results, that shows us that the experiment is reproducible. How could you improve precision? Well, to improve precision, we can increase the resolution of our measuring instrument. So for example, you can buy those wooden meter rulers that go up in centimetres. That wouldn't be any good for this experiment. What we want to use is a nice plastic transparent ruler, and to measure to the millimetre would be enough. So the other thing we can do is we can measure the height of the seedling, but then we can also measure the height 3 times, and then we can take an average then with those 3 measurements, and that will improve the precision. So I hope that's made sense to you. We're going to be doing more and more of these because ultimately in AQA, this is what they're going to be asking you to do in an exam. It's going to be a 6 marker, and they're going to be asking you what equipment you'd use, what range you'd use, what intervals, what resolution, the precision and the accuracy. So we need to get used to using all those terms and making sure that we know our variables.